Goodman, microbiologist with USDA in Adams, Oregon. And today I'll be sharing life underground, soil microbes, and nutrient cycling. I'll be describing some of the data that we're seeing in the long-term plots here at Pendleton. And the collaborators that I've been working with include both OSU colleagues and ARS. When I think of the soil, I imagine this interaction between the soil environment, so the chemistry, structure, temperature, moisture, and the microbes. So the environment can influence the soil microbes, their activity and their abundance, and in return, the soil microbes can change the chemistry with their activity, soil aggregation, their different diversity, and also abundance. An example of this interaction are with the diazotropes. These are the nitrogen fixers that take dinitrogen gas from the atmosphere and they convert it into ammonia in the soil. These organisms are inhibited by ammonium, one of the products of their process. So they're inhibited both on an enzymatic level and also on transcription, so taking their DNA and then getting the transcripts to be made into protein. This is data that Harrow and Stewart and I collected in 2014, and you can see in this panel we estimated the number of these organisms, the diazotropes, by looking at one of the gene targets involved in the pathway of nitrogen fixation. We were able to count that from the soil using quantitative PCR. And along the bottom you can see the two treatments. These are from the long-term experiments that are being managed by Harrow. There's 0 nitrogen, and this is in a wheat fallow system, and we're comparing that to one of the higher nitrogen rates at 160 pounds of nitrogen. So the nitrogen was banded into the soil at a 10 centimeter depth as urea. You can see the bars here in the top 2 inches of soil are no different between fertilized and unfertilized, but here at this banding depth, where the urea was applied, we see the strong inhibition of the diazotropes. And below that depth, we don't see that inhibition. The letters over the bars indicate that they're significant. So if they share a letter, then they're not different. But we do see that difference there at the fertilizer banding, suggesting that these organisms at this depth are being inhibited. So let's talk about the nutrient cycle. Nutrient cycling is this flux between the biotic and the abiotic pools. So in this example here with this tree, we can see that there are nutrients that are required for the tree growth. And then also we have deposition of some of these nutrients by the air and by rainwater, plant and animal litter. And then once these come into the soil, especially as complex nutrients, you have the decomposers that are going to depolymerize and break these organic materials down so the smaller nutrients can enter different cycles such as nitrogen cycling. When I talk about decomposition, I'm talking about the degradation of organic matter into simple and inorganic compounds. For decomposition or degradation, it takes more than 50 extracellular enzymes to break down a microbial cell or leaf litter. This is a large interaction of enzymes able to convert this complex nutrient down into simple molecules. You can see around this circle enzymes that are involved in carbon cycling, nitrogen cycling, sulfur cycling, and phosphorus. An example of this degradation is here with cellulose on the left. You can see these long chains of glucose molecules that with 3 different enzymes that are able to cleave at specific places in this long polymer is able to release glucose molecules. The enzymes that I'll be talking about are glucosidase, involved in the carbon cycle, beta-glucosaminidase, amidase, which releases amino acids from larger molecules, aryl sulfatase, and also phosphatase. You'll see 2 different phosphorus enzymes, alkaline and acid phosphatase. Be sure to remember these because they have different pH optimums. We were really interested to see what the long-term effects of nitrogen fertilization had on the soil community. So how that changed the soil properties, being the chemistry, and also how that influenced the soil activity for nutrient cycling. We've looked at the crop residue study, which Francisco Calderon and Stephen Machado had described in an earlier field day talk. These plots were established in 1931, and the treatments that we'll be looking at are 0 pounds of nitrogen, our unfertilized control, an inorganic fertilized treatment that received 90 pounds of nitrogen per acre per year, and also manure, in which it was applied equivalent to 99 pounds of nitrogen per acre per year. We sampled these during the crop phase, so we were able to sample both the rhizosphere, the soil attached to the root, and the bulk soil, which was impacted by the roots. When we looked at the enzyme activity of these soils, they were very similar in overall activity. Along the left is the sum of the soil enzyme activity, and you can see stacks of the different enzymes that we talked about before. Glucosidase, which is involved in carbon cycling, as well as glucosaminidase, involved in carbon and nitrogen cycling. Amidase, so releasing amino acids into the environment for nitrogen cycling. Sulfatase and acid phosphatase. Surprisingly, there wasn't a huge difference between the complex manure addition and the inorganic fertilized or controlled soils. But remember I said the environment influences the microbes and the activity, and they're able to change the environment. If we look at the soil pH, which is a large influence on the soil communities, we can see that there is a very significant difference between the control soil, 0 nitrogen, and the inorganic and manure treatments. These are very significant, a 10-fold decrease between unfertilized and fertilized soil, and a 100-fold increase between fertilized and manure fertilized soil. So this is often a very large factor driving the microbial activity and also the community composition. And remember I told you about the activity of acid and alkaline phosphatase, that they have different optimums? If we take a look back at that first graph and overlay alkaline phosphatase, you can see that the difference is much more significant for an increase in activity in the manure fertilized soil compared to the inorganic and control. I think that this highlights well how the environment can influence these communities and their activities. So stepping back to the big pictures, when you think about your soil communities, think about the environment. Think about the environment that the organisms are in, realizing that that environment, whether they have different chemistry from management practices or even different root deposits from different kind of crop species, that these can be factors and impacts to your soil. So as another part of the study, we've been looking at the community composition and the diversity of the organisms that are associated to the root. But unfortunately, I'm going to have to keep you on the edge of your seat until next field day to learn more about how the environment shifts the diversity and the community composition. Thank you.